Hello and welcome to the fifth topic in molecular biology, which is enzymes. And within this video, I'll be talking about the enzymes and the substrates, what they are and what their function is. Next, enzyme activity and what may inhibit this activity. And lastly, enzymes in industry. I hope you enjoy. So, enzymes in general are biological catalysts and globular proteins. And globular proteins, because of their shape, determines their function. So if you want to check that out, my last video is on proteins. And another specific thing about enzymes is that after their reaction with a substrate, they remain unchanged and may be used again. There are biological catalysts, which means that they speed up reactions by increasing the number of successful collisions. They are polypeptide chains folded into 3D shapes and are have a specifically shaped active site, a cleft, so to say, to which complementary specific substrates may bind. They are substrate specific according to the key log principle. And here you can see a picture of the key log principle, and a reaction goes generally as follows. The enzyme has this active site and the substrate is complementary shape to it, and through random movement they are brought together and a temporary enzyme substrate complex occurs between the R groups of the amino acids in the active sites, and they interact with their substrate. And in the third part, the interaction of substrate and enzyme breaks the substrate down, and they leave the unchanged enzyme to form other enzyme substrate complexes. So yes, this was the reaction, and enzyme activity in general is determined by the shape and chemical properties dependent on the tertiary structure of the protein. They can be modified and denatured by external factors, and one of those external factors is temperature, as you can see here in this graph. And they have an optimum temperature of about 37 degrees where they reach a peak, and afterwards they decrease, as you can see here. And low temperature results in insufficient thermal energy for activation of reaction, and a higher enzyme activity comes with rising temperature because of the higher kinetic energy and the higher percentage of collisions. The higher temperature decreases the enzyme stability after the peak is reached, so the thermal energy disrupts the hydrogen bonds and they lose their shape and specifically of the active site. This means that they have a loss of activity, so denaturation occurs, and that also means that they lose their biological function. Next up we have the pH value, and pH is measure of the relative numbers of hydrogen ions and oxyhydrogen ions in a solution. And a low pH, so an acidic pH value, means that there are many H+ ions and it's acidic. And a high pH means that there are many OH- ions, thus it's basic. The changing of the pH alters the charge of the enzyme, which alters the solubility and the shape of it, and the charged amino acid make bonds with the pH ions and thus inhibit the matching process between the enzyme and the substrate. And different enzymes have different pH optimums, therefore I cannot say anything on the optimum peak of enzymes. And lastly, we come to substrate concentrations as something that inhibits enzyme activity. And with increasing substrate concentration, there will be an increased activity of the corresponding enzymes. And more substrates means an increased chance of an enzyme and a substrate colliding and reacting, but after a certain point the rate of activity will cease to rise regardless of any further increases in the substrate levels, because the enzyme is saturated with substrate and all enzymes react. So we have the maximum reached, and we also see a plateau here, because it isn't really the speed of the enzyme reacting that's changing, but just the rate at which they meet with substrates. Exactly, and this was on the change of enzyme activity. Next up, we move on to enzymes in the industry, and we have For example, immobilized enzymes that are fixed to a static surface to improve efficiency. And those enzyme concentrations are conserved, and there's an easy separation of the product. An example in the industry may be biofuels, which is the breakdown of carbohydrates to produce ethanol-based fuels. We also have it in medicine, which is to identify a range of conditions. And then we also have biotechnology, for example, gene splicing. Food production, wherein the production and refinement of beer and dairy products occurs. Textiles to process fibers, paper, that includes the pulping of wood for paper production. And there's also a special case in the food production, where we can produce lactose-free milk with the enzyme of lactase for lactose intolerant or vegan people, And this was it on enzymes. I thank you for watching and listening. Hope you have a nice day. These are my sources.